When we talk about the cell, we went real quickly to the cell membrane. I told you, don't worry, we are going to cover. So we have a whole video devoted only to the cell membrane. That's how important it is. Remember, the cell plasma membrane or cell membrane defines the boundary of the cells and determines how it interacts with the environment. It will allow certain things to cross, it allows certain things to come in, or even carry them forcefully inside. It will excrete things that the cell wants to get rid of. So the plasma membrane is dynamic, is constantly changing, it's very flexible, and something very important we mentioned, carries markers. Remember those carbohydrates? We did look at it in the previous video, that allow the cells to recognize each other. So you get infected by a cell, by bacteria, they're going to recognize those markers. You get a transplant, they're going to identify that, hey, this heart does not belong to this body. We already talked about that. And the plasma membrane carries receptors. They're very specific attachments for some substances that interact with the cell. Basically, cells can send chemical messages to other cells, some of them very far. Let's imagine you ate breakfast, okay? So your sugar is high. Let me guess. You eat breakfast, so your sugar in blood goes up. What happens? Your pancreas responds to the high sugar by making a chemical that we call insulin. That insulin goes into the blood, is expressed throughout the whole body, but only the cells that have receptors for insulin, for instance, muscle cells, liver cells, those bind insulin and respond by uploading the sugar and removing it from the blood. All cells are exposed to insulin in your body, but only the ones that have receptors respond to it. What we know about the plasma membrane can be explained by the fluid mosaic model. First is a model, obviously. Mosaic means that it's made up of many different components. You can see that in there. And fluid, we talked about that earlier in the previous video, how some of these components can move. Okay. So we talk about phospholipid bilayer. We talk about how the hydrophilic phosphate heads are sticking to the outside of the membrane. That can be the outside or the inside of the cell in there. The hydrophobic fatty tails are hidden inside. You have proteins that are going to have a bunch of different functions. Some of them cross the membrane and they're called integral. So they're usually the transport proteins or receptors. Some stay to one side or the other, it doesn't matter which one, but they're called peripheral. And they can be enzymes, they can be the proteins that have the carbohydrates that we use to identify. We did talk about cholesterol as maintaining the right fluidity. So the plasma membrane is really, really complex. So how do things move through the plasma membrane? Okay. We say the plasma membrane allows certain things in, but not others. So we say that it's selectively permeable. When a molecule wants to cross the plasma membrane says, okay, that's fine, go in. Or, no, we don't need you, and you're not going to be able to cross. Now, there's two different types of transport. Active transport, it means it requires energy. If you're going to be transported in your car, it requires energy, right? You have to burn gas to do that. In this particular case, since we're talking about cells, it's going to be ATP. And then you have passive transport, where no energy is required. And we're going to be looking at the different types of both of them. Okay? So passive transport, we said no energy required. Basically, a substance moves down the concentration gradient. Imagine you have a glass of water and put a drop of food coloring. Basically, the food coloring is very concentrated in that drop, and there's none in the water. So if you wait long enough, what happens? The whole water turns the color of the food coloring. This is spontaneous. It didn't use any energy. It just, the molecules of food coloring move from the high concentration to the low concentration. There's 3 types of this passive transport. Can be simple diffusion. This is the case we saw in there. It just happens. Facilitated diffusion means that needs help. And then osmosis, that's going to be movement of water. And let's look at each one. Simple diffusion is, again, the particles move through the membrane down the concentration gradient. So you have the membrane in here. It's a particle that can dissolve in the plasma membrane. So there's a lot outside. There's none inside. So slowly they started seeping through it, fusing through it, until it equalizes the concentration on both sides. How fast they diffuse? You can influence it because the smaller the particles, the faster. I mean, after all, they're going to have to squeeze in between the different components of the plasma membrane. And the warmer it is, the more energy the particles have, so the faster they move. But there is no help in here. It's just going down a concentration gradient. Facilitated diffusion is for those molecules that don't dissolve in the lipid. Remember, the phospholipid bilayer has this part in here that's lipids, is very hydrophobic. So hydrophobic molecules dissolve very easily, no problem. But if you have something like glucose that's very polar, or ions that have charges, they're not going to cross the hydrophobic part of the plasma membrane. So how do you get them in? You have integral membrane proteins, carrier proteins, channel proteins. Basically, when the channel opens, they're still diffusing down the concentration gradient. Down concentration gradient, that's what's called diffusion. But they need help, they need the protein to open the channel. That was called facilitated. Osmosis. Osmosis when water is going to diffuse. Up until now, we were talking about the molecules dissolving the water, but now we're going to be looking at the water itself. And it's going to diffuse from high water concentration to low water concentration, because that's what diffusion is. Now, don't get confused, because people confuse regular concentration with water concentration. Let me give you an example. I have one solution that has 50% glucose, and another one that's just 5% glucose. And you can tell me, oh, this is the most concentrated. Yes. But if we're looking at the water, if 50% is glucose, that means it's 50% water. If this is 5% glucose, that means it's 95% water. So the water concentration runs opposite the solid concentration. Okay? In osmosis, we're talking about the water. So osmosis will take place from the 95 to the 50. If we was looking at diffusion, it would go from 50% glucose to the 5, but we're looking at the movement of water. Okay? If you put a semi-permeable membrane, that means the solid cannot cross, and then the water's the only one that can go in there. So water's going to be moving across the semi-permeable membrane, trying to equalize the concentration. Of course, it can't, but you can see the level increases dramatically in here, because water's moving from the left to the right. At a certain point, this column, the weight of this column is going to be pushing down, so you have a force trying to move the water to the right, and we have a force, the weight of the water moving to the left. Eventually you reach an equilibrium. Okay? In the case of the cells, the water can cross directly through the lipid bilayer. Yes, it's a polar molecule, but it's small. So small amounts of water will diffuse to the plasma membrane, rather osmosis. But we have also water channels, aquaporins, that allows larger amounts of water to go through. And certain cells are very good at making and removing aquaporins. For instance, the cells that line the tubules in your kidneys, they need water moving in and out a lot. So they're going to have lots of aquaporins. The concentration of solid particles in a solution is called osmolarity. That comes from a chemistry term. It's trying to talk about osmosis, but molarity is a term for concentration in chemistry, so come up with this. But basically, the more solute, the higher the osmolarity. When you have two solutions with different osmolarity in a semi-permeable membrane, the water can move through the membrane and osmosis takes place. Now, when you compare the concentration of a solute to that of the inside of the cell, that's what we refer to as tonicity. So there are three different possibilities. Hypertonic. Hyper means high, more than. So a hypertonic solution has a greater solute than the cell. Hypotonic, hypo means below, so that has a lesser concentration than the cell. Isotonic, iso means the same, so that has the same solid concentration. And this is going to be critical for cells, okay? Let me give you an example. What happens if you put red blood cells into three solutions? One is hypertonic, one isotonic, one hypotonic. Okay? Hypertonic means that there is a lot of solute outside. So let me call it, there's a lot of solute, which means that there's less water. So there's more water inside than outside. By osmosis, the water is going to be moving from the inside, to the outside. Water comes out, the cell shrinks. Actually, it eventually, that's called crenation, by the way. And if the cell loses enough water, it will die. In an isotonic solution, you have the same amount of water inside and outside. So yes, water is going to be coming in, but it's going to be coming in and coming out at the same rate, so nothing changes. Now, in a hypotonic solution, we have very little solute, which means we have a lot of water. More water in the outside than in the inside, so water comes in to the cell starts swelling, and eventually can burst. This is very important. For instance, if you give an IV to it better be an isotonic solution, because you can literally kill that person if your IV is hypertonic or hypotonic. You're killing the cells either way. This will happen with animal cells that don't have a cell wall. Plants, they're not going to shrink completely, they're not going to burst, but the turgor pressure, that means the pressure inside the cell, is going to depend on the tonicity. Hypertonic, the water comes out. Well, the cell is not going to shrink completely because it's attached to the cell wall. You can see these projections in here. So yes, it shrinks a little bit. Your plant wilts. All of a sudden, the leaves are falling down, and you say, "Oh, I forgot to water the plant," and you water it, and everything's fine. Isotonic, we said there's no change. Hypotonic, you have water coming in, but the cell is not going to burst. Why? Because the cell wall is protecting it. Okay? So that's a big difference. We're talking about animal or plant cells. The other form of transport we talked about was active transport, and we say it requires energy, that in the cell obviously is ATP. So why do you need energy? Because you're moving against the concentration gradient. Basically what you're saying is, on this side of the membrane, I have only one molecule. On the other side, I have a bunch of them. If these particles could cross the plasma membrane, they would tend to move from high concentration to low concentration, but you really want to move this one up. You're going against the flow. You're going to have to give energy. I like to always compare active and passive transport as, imagine you have a little hill covered with snow, and just a little hill, and you have a little red wagon. If you push it, the red wagon is going to go down by itself. You don't have to give it energy. But if you enjoy and you say, "This is fun, let's do it again," and you have to pull the red wagon up the hill, now you're going to have to work, right? You're going up the hill, pulling on the wagon, that uses energy. One term you're going to find a lot in biology is electrochemical gradient. Basically, it's not only a concentration gradient, but it's a concentration and an electrical gradient. Okay? And these electrical gradients are going to be very important when we talk about muscle and nerve cells. But it's the same thing. you're going to be moving particles against the gradient, so you're going to have to use energy. Here you have a nice example, and you can see how you have very little chlorine in here. So the concentration gradient is going to move it in this direction, but you also have to keep in mind the charges. Okay, so you're going to have some positive charges moving in this direction. Why? Well, because you have these negative charges in here that are going to attract them. So you're going to have many times to consider both the concentration gradient and the electrical gradient to see in which direction a particular particle is going to move. Fortunately, to keep things right in the cell, because we said this is critical for nerve and muscle, you have a pump. Anytime you have a problem that if you provide ATP, a transport solute against the concentration gradient, that's a pump. Kind of like when your tires are low, what do? You pump air into the tires. You don't necessarily have to have a pump. You go to gas station, but you're pumping air in there. Cells have a very important pump, the sodium-potassium pump. You will learn that the sodium and the potassium are going to be moving in and out with the transmission of nervous impulse, with the transmission of the signal to the muscle, something like that, and you need to regenerate your original status. So you have this potassium pump that pumps potassium in to maintain high potassium inside the cell, while pumping sodium out to keep high sodium outside the cell. The potassium is already high inside, the sodium is already high outside, so it needs energy. And notice that the number is not equal, but it uses ATP to pump. This is a very important pump. Without it, as the first impulse in your nervous or muscle system, your cells will be out of commission. So this is a very important pump. We talk about vesicular transport. What if you have something really big or a macromolecule, something that you want to go across the membrane? Well, the solution is to surround it by a membrane, so you make a vesicle, and to get it out, it's going to require ATP. This is active transport. So your Golgi apparatus just package something that's going to be secreted outside the cell and is sent to the membrane. So there's basically two ways big particles can cross the membrane. It can go in or it can come out. So endocytosis means transporting materials into the cell. Exocytosis is out of the cell. Basically, endocytosis means the cell is taking something in. If it's taking solids, it's called phagocytosis. Phago means to eat, the cell eats. Pinocytosis means taking in fluids. Pino means drink, so the cell is drinking. And then there is a receptor-mediated endocytosis that allows receptors to target what things are going to be taken into the cell. Okay, and we're going to look at an example in a minute. Exocytosis, on the other hand, means, like I said, transporting materials outside the cell. So here I'm trying to show you phagocytosis. You have a large particle, maybe it's a bacteria because you're a white blood cell, doesn't matter. The plasma membrane surrounds it, makes a vesicle, and the particle comes in. If instead of a solid particle it's liquid, that's called pinocytosis, but it's the same idea. The plasma membrane surrounds it, makes a vesicle, and then the vesicle. Now, suppose that all I want to get is the little stars. Okay? If I did phagocytosis, I'll get the stars and the circles, but I only want the stars. If you have receptors, the receptors are going to bind the stars. So then, when the plasma membrane forms a vesicle, it didn't take everything that was around. It only took whatever the receptor had caught. So it's endocytosis, but it's held by the receptors, so you only take into the cell a particular molecule that the cell is interested in, instead of everything that's around it. Exocytosis is the opposite process. So you have a vesicle, maybe it comes from the Golgi, you just make some proteins, Like mine is the mammary gland, and it's making milk, and these are the proins from the milk casing. That vesicle is going to fuse with the plasma membrane. When it fuses, it opens, and that proin comes out. That proin is too big, it could not cross the plasma membrane, but notice that when this fuses and now it separates, it didn't have to cross. The vesicle becomes part of the plasma membrane, so the proins went from the inside to the outside without having to cross the membrane. Isn't that amazing? So, I know this has a few more terms and all of that. Once again, you want to work on a one-to-one basis, you want to not have problems for biology, but you want to look at any of the charts summarizing a particular chapter, or you want to start for an exam, go to my Washington profile and make an appointment. You have a link to my Washington account, make an appointment, and I'll be glad to meet with you.